In this video we will talk about the genetic disorders involving mitochondria and therapy. Let's discuss the genetic disorders involving mitochondria first. Mitochondrial DNA-associated Leigh syndrome, which affects ATP6. You can remember it by a 6. Moving to next genetic disorder involving mitochondria. Mitochondrial myopathy, encephalopathy, lactic, acidosis, stroke-like episodes syndrome. Affects transfer RNA, leucine gene, and cytochrome oxidase of complex 4. You can remember it as 4. L indicates for leucine, and 4 indicates for cytochrome oxidase of complex 4. Moving to next genetic disorder involving mitochondria. Myoclonic epilepsy with ragged red fibers syndrome. Affects transfer RNA leucine gene and NADH, dehydrogenase of complex 1, and cytochrome oxidase of complex 4. You can remember it as 1 for K for leucine, and 1 and 4 indicates for NADH dehydrogenase of complex 1 and cytochrome oxidase of complex 4. Moving to next genetic disorder involving mitochondria. Leber's hereditary optic neuropathy. Affects subunit 4 of NADH dehydrogenase, subunit 1 of NADH dehydrogenase, and subunit 6 of NADH dehydrogenase. You can remember it as this, as subunit 1, 4, and 6 of NADH dehydrogenase are affected. Moving forward, mitochondrial mutations may affect human aging. Some mutations in mitochondrial DNA are inherited through the germ line, while others arise sporadically in somatic cells as a result of random events such as exposure to radiation or chemical mutagens. In fact, in humans the rate of somatic mutations is much higher in mitochondrial DNA than in nuclear DNA. One reason is that the mitochondrial oxidative phosphorylation system generates high levels of DNA, damaging free radicals within the organelle. Some researchers focusing on the genetics of aging think that the accumulation of mitochondrial DNA mutations over a person's lifetime results in a decline in oxidative phosphorylation. This decline, in turn, accounts for some of the symptoms of aging, such as decreases in heart and brain function. One piece of evidence supporting this hypothesis is that the brain cells of people showing symptoms of Alzheimer's disease have an abnormally low energy metabolism. Now let's discuss the therapy. Well, oocyte nuclear transplantation can sidestep transmission of mitochondrial disease. Mitochondrial disease is relatively rare, yet every year in the United States alone, several thousand babies are born with a disease caused by mitochondrial DNA mutation. New technology may soon make it possible for an affected mother to avoid transmitting the mitochondrial disease to her children. The idea behind this technology referred to as mitochondrial gene therapy. So, let's understand mitochondrial gene therapy. Mother's egg having mutated mitochondria. So, nucleus is removed from the mother's egg. Donor's egg having normal mitochondria. So, nucleus is also removed from the donor's egg. The resulting eggs, which have nuclei from one source and mitochondria from a different source, are called hybrids. So, after in vitro fertilization, the zygotes can be implanted in a womb. The resulting child would have the nuclear genes of his or her parents and the normal mitochondrial DNA of the egg donor. In 2009, a team of researchers transferred the nuclei of rhesus monkey eggs into enucleated donor eggs and fertilized these manipulated eggs in vitro with rhesus monkey sperm. The zygotes were implanted into a surrogate mother's womb. Apparently nonal fraternal twins, Mito and Tracker, were born, and molecular analysis showed they had mitochondria DNA, only from the cytoplasmic donor. The success of the monkey experiment led the government of the United Kingdom in 2015 to approve clinical trials of mitochondrial gene therapy in humans. As of this writing in 2016, the US government is still debating the ethics of this new technology. Well, hope you have a better understanding of the mitochondrial inheritance. Thank you for learning.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.